This video is going to be about protein monomers, which are amino acids. And remember from our chart, when we were talking about the elements that were present in proteins, we had carbon, hydrogen, oxygen, and nitrogen. And you'll be able to see all of these in an individual amino acid. So let's make a sketch of one of these amino acids. The backbone of an amino acid is going to be a nitrogen, a carbon, and a carbon. So already, boom, got our nitrogen covered, got our carbon covered. Over on the left side here, attached to our nitrogen, is going to be two hydrogens. So we have our hydrogen covered. Then on the right side, attached to our carbon, the farthest carbon over, is going to be a double bonded oxygen and an OH group. And now we have our oxygen. So we've covered all the elements that we need in a protein. But now let's go into a little bit more depth about what we have going on here. So this nitrogen over here that's connected to two hydrogens, we call that our amine group. And that's where you get that name amino acid from. And then over here, attached to our carbon, with the double bonded oxygen and the OH group, we call that a carboxyl group. That's not all though, because attached to the center carbon here is a hydrogen on the top, and we know that carbon, because it has 4 electrons in its outer shell, can make 4 bonds. And right now we have 1, 2, 3 bonds. So this carbon is going to be bonded to one other thing, and we call that one other thing the letter R. We say this is the R group. R in this situation here is just a variable, because when we think about amino acids, we have to remember that there are 20 different amino acids. And sometimes when you see me abbreviate amino acids, I'll put 2 capital A's together. There are 20 different amino acids, and they're different because they have different R groups. So each amino acid has its own R group. These R groups can be split up into classes. So amino acids are split into classes based on these R groups. So you may have some R groups that are nonpolar. Remember, those electrons are being shared evenly. Some R groups may be polar. And some R groups may be electrically charged. So the difference between having an R group that's nonpolar versus having an R group that's polar is that the polarity of an R group will affect solubility. And remember from our water chemistry unit that solubility just means the ability to dissolve in water. And then if it's electrically charged, what that means is that you may have positively charged and you may have negatively charged R groups. If an R group is positively charged, it most likely attracted a hydrogen ion, so a positively charged hydrogen, and therefore is basic as far as pH goes. If an R group is negative, it probably had gained a hydrogen ion from whatever solution it was in and is therefore acidic. So the different classes of R groups affect how those amino acids are going to act in solution.